Hi children, welcome back to this ELISA test. Here you are going to learn about the indirect ELISA test. This is the best method to identify the HIV. In this test, the secondary antibodies are used. In the direct ELISA test, primary antibodies are used, but in the indirect ELISA test, secondary antibodies are used. Here we'll discuss about the protocol and how the test is conducted. Here blood is collected from the HIV person and the serum is gets separated. This serum process with antibodies is applied to the titer plate. These known antigens are attached in the wells of the titer plate. Now the antibodies are given, or now the serum is applied in this titer box, so that the antibodies present in the serum are attached to this known antigen of HIV. So what will happen? These antigens may have multivalent sites, antigenic sites. So number of secondary antibodies may allow to attach. Now we have to apply the enzyme-linked anti-human immunoserum globulins to this titer plate. These are now termed as the secondary antibodies. These are the secondary antibodies. These are called as the antibodies. These secondary antibodies now supplied with the substrate. What will happen? One antibody is attached by three secondary antibodies, enzyme-linked antibodies. So naturally substrate is also attached with three antibodies and reaction takes place at three sites. So what will happen? The color will change more as compared with the direct ELISA test. So this is a more sensitive test and more specificity is identified. So we can easily identify the antibodies by this indirect ELISA test. How these secondary antibodies are prepared? Generally, if it is HIV person, the antibodies, the serum is collected with the antibodies. These antibodies, for example, given into the testing organisms, for example, rabbit. In those organisms, these antibodies produces again antibodies, anti-human immunoglobulins are produced. Anti-human serum immunoglobulins are produced. These are called as secondary antibodies. These are linked with enzyme and supplied. So these are now called as enzyme-linked anti-human immunoserum globulins. So these react with the substrate and changes the color. If you observe, compare direct and indirect, observing that only one enzyme-linked antibody is attached with this complex. Here, three enzyme-linked antibodies are attached with this complex and get reacted. So, this color produced here is less, and the color produced here is comparatively more than this. We can get more specificity here. If there is no anti-HIV antibodies in the serum sample, there is no binding of primary antibodies to the antigens. So, there is no color change. So, the test is said to be negative. Sometimes it may be false positive under certain circumstances, and also false negative under window period. So by using this technique, we can identify the test for HIV. Question for you is how the secondary antibodies are produced. With this ELISA test, our syllabus is completed. I hope you have understood well. Thank you.